Okay, so this is an overview of what we did for the AI-powered analysis Pathfinder, and I should say that's been tightly linked with the RIA MCP group as well. What we've worked on that was a bit serendipitous was this collaborative use case that Jonathan Livney brought doing analysis of various studies around Pseudomonas. And since BRC Analytics is the new kid on the block, we are going briefly explaining what we're using, so what this is all about. In terms of resources used, we have used workflows from the IWC, that's the Intergalactic Workflows Commission. So that's a community resource of high quality and curated workflows. And people can just go there. There's currently 105 workflows. They all have descriptions, tests, all the good stuff. And then we used the Galaxy Training Network. It has Galaxy in the name, but it's not just Galaxy. And that contains maybe the largest amount of training materials in any given specific site. And there's a huge amount of resources for scientists to learn about an analysis. And it's linked to Galaxy, but not exclusively. And that's a great resource to figure out what you want to do and run. And then we have a couple other sources as well. And so we have a embeddings database that is constructed from all of those sources to make them easily searchable in the MCP context. And we're also going to add the tool embeddings from the RIA MCP group. So we're going to hear about that after this presentation. So we have the MCP server we have worked on, and there are prototypes for an MCP server in Galaxy, as well as an external MCP server that you can, as a user, interact with. And we plan to build nice integrated pieces, that agents that you can combine. There's various MCP tools already. So for this one, we're going to show like just a small bit that we've used. So here's the external usage. So it's a MCP server running locally, and these tools are all interacting with a remote Galaxy server. So this is running against a production instance on usegalaxy.org. And I think this is probably too small to recognize anything, certainly from here. I can't read, and I don't remember exactly what I wrote, but I can give you a rough gist. So I've asked, I want to run an RNA-seq workflow. How do I do that? And then the MCP tool kicked in. And it said, yeah, I know about this. I said that I wanted to do an RNA-seq analysis. So it started searching for workflows. So that's here. And these are all individual tools. So we have a connect tool, we have a create history tool, and we have a search workflows tool. And so this search currently looks at what workflows are available. It's not yet using the embeddings. So it found an RNA-seq workflow that I asked for. It lists all the major steps that it's doing, and that's great. And we have the Pseudomonas data provided by Jonathan's group. And then the next step is like the workflow describes what kind of inputs it requires. So we need, of course, the reference genome. We need the annotation files to do gene level summary. And all of these were being found and directly uploaded to Galaxy. So there's nothing running locally. It's all just URLs plugged directly into Galaxy through the MCP tool, and they appear in the history. So yeah, you can see here that these files are uploaded from a URL. And then, yeah, the next step is, can you run the workflow? And unfortunately, the answer is no. We've had it working, but I don't really know what broke. However, in principle, this works. We can probably fix that up real quick. But it attempted to. It invoked the tool, but the tool itself failed doing double JSON encoding somewhere. It's a little sad, but that's how it is sometimes. And so in the BRC context, the goal is really to, in natural language, have researchers describe what they want to do. And they don't necessarily always know like what are the exact taxonomic IDs. That's a hard one. So we developed also a Claude skill to translate like things like rat and black plague back into like the taxonomic IDs. And then we also developed a skill that can recommend workflows. So for instance, like, yeah, if I want to do surveillance analysis, which workflows would you suggest? And suggest pathogen genome sequencing for strain typing, variant calling to track mutations, phylogenetic analysis, and maybe metagenomic analysis. And that all makes sense. And then you can get the individual IDs. So that should be a nice building block to build like a nice integrated experience. So this is now a quick demo to show you what we can actually do with this. I've basically stated in plain text one of the questions you can ask on the data, and from a very naive point of view, where I'm asking, I want to know about the differential expression between two CRISPR-I conditions in a specific Pseudomonas strain. And can you suggest the data in the workflow? So then the LLM is thinking and thinking and thinking a little more. And it suggested two workflows. And these are actually the RNA-seq workflows for single end and paired end. Did it find the data as well? Cool. Yeah. And so actually the first project contains the data we're interested in. Like I asked for differential expression between genes, and I got the right data and I got the right workflow. And the next step is then setting this all up so that you can do like the contrasts between the conditions to run the whole workflow front to end. We also did a little bit of non-AI things to first do the manual portion of it. So for instance, a lot of sequencing centers work with sample sheets, and we've sort of prototyped the sample sheet approach for that same data. And all you got to do is like find the workflow you want to run and upload that sample sheet. And the sample sheet then specifies the replicates and the treatments so that you can form the contrasts that you actually want to use. And we're also then planning, because this is a specific sample sheet that's not directly corresponding to the metadata you can get from the sequencing read archives, there's going to be like an intermediate LLM connector to morph that into exactly the right shape, or give a best guess at what it could be doing. So then as a researcher, you really don't need to worry about getting things in, understanding concepts. You can just click go, which is already a big advantage. But of course, ultimately the analysis is to finish this. And then finally, because like things are all about generative workflows, we've run a workflow that already existed, but I want to just briefly show you the super powerful new thing that we have in Galaxy, where you can describe a tool that is supposed to do something. So in this case, it's supposed to like 10 lines from a file, user-specified number of things from a file, and it generates that new tool. So that means you can interactively run an analysis, and you being you as a human or an agent. And Galaxy has already this feature built in to extract workflows back out from an interactive analysis. So what's happening here is that we've generated this tool. It runs containerized. It runs on big compute infrastructure, whatever is configured for the Galaxy instance. And there's really no limit. Like you can run all your command line things, so you're not stuck in any way. That's it.